everybody, and welcome to another episode of In Plain English. I am your host, Jamie Moffa, and today we are going to be discussing the paper 5-MeO-DMT modifies innate behaviors and promotes structural neuroplasticity in mice by Sarah J. Jefferson et al. Here to discuss this paper with us is our expert guest for today, Alex Kwan. Alex, would you like to introduce yourself?
Yeah, I'm glad to be here. So right now I'm an associate professor in the Lanick School of Biomedical Engineering at Cornell University, which is in Ithaca, New York. My lab has recently, several years, have been interested in research in psychedelics. I'm glad to discuss the paper here.
Yeah, we're glad to have you. And then joining us as our layperson guests are Raven and Asmodeus. Would you each like to briefly introduce yourselves?
You want to go first?
I'm Asmodeus. I am half of the Smoking Out the Closet podcast, where we talk about cannabis and the LGBT community.
I'm Raven, the other half of the podcast. We also work in the cannabis industry.
Great. Well, without further ado, let's get into the paper.
Well, first of all, I want to mention that this is not a published paper yet, but this is what's known as a preprint, which means that we have completed a study and we've written up the results and we put it up on a preprint server. So it's shared with the public, but on the other hand, it hasn't gone through the full peer review yet, which will surely change the paper somewhat. Although again, it's good enough that we thought we can share it. So the premise of the paper is that a couple of years ago, my lab has another study showing that if we give the mouse a single dose of the psychedelic psilocybin to mice, then we can observe some noted neurite outgrowth or growth of dendrites, which is an indicator of the amount of neuronal connections in the brain. So we show that by injecting mice with a psychedelic dose of psilocybin. And then later on, we use optical imaging to track these neuronal connections, which are known as dendritic spines in the brain over time. And then there we found that it has a fairly rapid effect and a long-lasting effect on the dendritic neural architecture within the brain of the mouse. So then following up the study, one of the questions that we have is that could another related type of classical psychedelics have this similar kind of effects on neuroplasticity? And then, moreover, we were quite intrigued by the compound 5-methoxy-DMT because 5-methoxy-DMT has some different characteristics than psilocybin, even though they're both classical psychedelic. One of the key differences is that it's a much shorter acting psychedelic. So there's certain advantage for having this type of compound where if you eventually want to bring it to clinic, you might want an action to be shorter. And moreover, chemically, it's also quite different. They target with a certain bias over some subtype of serotonin receptor that are quite distinct to psilocybin. So that's sort of the starting point on why we wanted to test the effects of 5-methoxy-DMT in mice.
Can you actually give a little bit of an overview of like what you mean by neuroplasticity and why is this concept important for what you're studying?
Definitely, yeah. I think that's actually a good starting point. I think take a step back to look at the bigger picture. Psychedelic recently have received a lot of attention as potentially being useful and beneficial for treating mental illnesses, particularly depression. Then these are longer lasting effects that typically arise on the orders of days and then maybe last quite a long time as well. Pilot clinical trial has shown that the benefits can last for on the orders of weeks to months. And these are timescales that are quite distinct from what's actually the more commonly associated effects of these compounds, which is the psychedelic trip, which is much more acute. For psilocybin, that happens between anywhere from 3 to 6 hours. So there's really a mystery here on how a drug that have these really acute, quick effect that lasts for 3 to 6 hours, and then the drug is gone from the body, can then elicit some longer benefit for mental illness. And I would say one of the prevailing and leading theory right now is that perhaps in addition to those acute perceptual effects and cognitive effects, these compounds could also enhance neuroplasticity in the brain and then leave a lasting change in the brain circuitry that then is what drive those beneficial effects. One way to look at that would be to go and directly measure the presence of these neural connections, which is something that we can do in the lab by imaging neurons at cellular resolution. Yeah, so I think this is, a good introduction to why I think plasticity is an exciting area of study for psychedelics.
Now, I do have a question on the effects of the DMT, because I was trying to read the paper, and to me, it read more like radio instructions. It was kind of confusing. So if I recall correctly, with the paper itself, it was saying that the DMT had both a short onset effect, but also a short, lasting effect on mental health. If I read that correctly, or am I backwards on that one?
So this is a good point, and this is, I think, an unknown right now in the field, and it's a big debate and why we want to do this study. So as I mentioned, the 5-methoxy-DMT has a much shorter time course. So psilocybin, 3 to 6 hours, but if one was to smoke 5-methoxy-DMT, the effect could arise within minutes, and it could be gone within 20 minutes. So that acute effect is much shorter. And people describe it as something like, oh, I'm being strapped to a rocket and blast off to space. And that's one type of description about that effect. It's just that rapid and that kind of fleeting. So the open question is whether, if you have a short acute effect, whether some of the plasticity outcome might also be short. Are those things linked? Or maybe there's a scenario where they're not linked. Maybe the duration of that acute effect and the duration of the benefit are not coupled. So that's actually an open scientific question, and one of the also motivations of why we want to do this study. Because if you push it to the extreme, there's also now a lot of companies trying to invest in the so-called non-psychedelic but serotonin 5-HT2A agonists. Can you even completely remove the trip, to which I would then say the acute duration is 0, but still maintain that mental illness benefit? So that's one of the things we also want to test here, whether a shorter acting compound, whether that plasticity effect we can see is also short or long.
Okay, cool.
And then before we go further, I also just want to tell that this is a common misconception. A lot of people, so DMT is actually a different compound than 5-methoxy-DMT, even though the names are very similar. DMT is the commonly more associated with as an active ingredient in the ayahuasca from South America, or the brew that you can drink. Where 5-methoxy-DMT is a different compound, most commonly found in the Sonoran Desert toad that live in Arizona, New Mexico, and northern Mexico. They have different sort of properties. They're actually both short-acting, but they have different types of effects: perceptual effects, cognitive effects, emotional effects on humans.
That's good to know on that side.
I have a quick question. I assume you can synthesize the 5-methoxy-DMT rather than having to go out and find toads?
This is a very, the whole toad story is fascinating. I don't think I can retell it to its justice, but if you watch Hamilton Pharmacopoeia, he has 2 to 3 episodes on 5-methoxy-DMT and how they, discover it and the whole history behind it. But the natural source there's several natural sources, but the most prominent one is the toad. And then to do it, what you need to do is part of in the venom, right? It's in the toad's gland. You have to squeeze it, and then it shoots out these white substances that you then dry, and then you would smoke it because I think it gets rid of the toxin. But on the other hand, it also can bypass your first pass metabolism in your body in order to get the drug into your head. Yes, there's actually a big conservation effort right now to not capture the toad to get the 5-methoxy-DMT because the toad are dying. Even though the process itself doesn't necessarily kill the toad, but a lot of people end up killing the toad doing this. So yes, I fully support synthesizing it. You also know how much there are. You can weigh it. It's much more pure. You should definitely do that. Moving forward, one of the first thing that we did in the study then, given this background and the interest in this compound is there's not a whole lot of actually study of this compound in animals. I think a lot of the study in basic research has been focused on other psychedelics like psilocybin and LSD. So the very first thing we did was actually give these compounds to mice and observe a fairly classic behavior output that the mice would do when they receive a psychedelic compound, which is called the head twitch response. So this is a very well-known phenomenon since the 1960s, where if the mouse received psychedelic, they would twitch and move their head very rapidly to the order of about 90 times a second. This very quick movement of the head is is a natural response, so you don't have to do anything to the animal to get this response. And it really showed that this compound is psychoactive, and it allows us to measure a dose response curve. If you give the animal different doses, you can watch this behavioral response to learn that, this is the dose where we can start to elicit a psychoactive effect in the animal. So that's one of the first things we did. And then to measure the head twitch here, we use a system that's initially developed by Javier Gonzalez Mizo at Virginia Commonwealth University, where we put a magnet on the mouse's ear. Anyway, this is a magnet-based system where we put the animals in an electric coil. So when the animal's head moves and the magnet moves, then it generates current. So it allows us to automatically and electrically detect the head movement. And then, the experiment that we did first was to give the mouse different doses of 5-methoxy-DMT to see how it changes the timing and the number of the head twitch response. We also gave it as a control, just vehicle, so nothing. And then we also gave it a dose of psilocybin that we know would work. And then look at how these different concentration of the drug would affect the mouse's behavior. So as you can see, one of the things that we found, two of the key point here is that one is that, yes, if we give more and more 5-methoxy-DMT, you see that the mouse twitch the head more and more, suggesting that the drug is dose dependent and psychoactive. And then the other thing that we see is that if you compare to psilocybin, no matter what dose we gave, the 5-methoxy-DMT always have a shorter duration of action. So what that means is the mouse will twitch the head, but always for a shorter period of time than if we give them psilocybin. So it seems like even in the mouse case, this is related, right? So in the human case, the effect is a lot more brief for the human, for the psychedelic trip of 5-methoxy-DMT. Same thing for the mouse. They also twitch the head, but for a much shorter period of time.
Now, with the whole theory of with it working towards, or the hypothesis, I should say, of it working towards mental health, are you guys looking into that with the mice study at all, or is it just a next step after this paper has been made?
Yeah, it's a good question. Another motivation for this study to start is that there has been a couple of clinical trials starting now for 5-methoxy-DMT. As I mentioned, it has some attractive properties, particularly because of the short duration of action, just a short trip. So if you need to go into clinic to receive this drug, the time that you need to spend there is a lot shorter than, say, psilocybin, where you have to spend several hours there at least. So there's a couple companies, including GH Science over in Ireland, and then Beckley Psytech is in the UK, is another company that's starting to test this drug. And the reason is there's also a lot of there's a good amount of recreational use of 5-methoxy-DMT. And then there are anecdotal report from some of those uses that the drug could also potentially be useful for if a person is having depressive symptoms. So there has been at least one phase 1 clinical trial completed to show that the drug is mostly have a decent safety profile. And now the ongoing phase 2 clinical trial to test primarily for depression. In my lab though, the question you asked though is whether, my lab's research, how does it relate to the clinic and whether it can translate. I see it as two potential segue there that would be helpful. One is that eventually we'll get to when do these drugs elicit and seem to enhance neuroplasticity. So if we know that time period and when the plasticity enhanced, then that might inform clinical work on when might be the best time to have assisted psychotherapy to further integrate that type of experience that arises from the enhanced plasticity. So inform the time period to which the drug is maybe exerting its beneficial actions. And then the other use I see for this type of research is that we can potentially use what we find in the brain as a further biomarker. If we know that this is what this drug does, we can find and evaluate other compounds that might be a better drug with even fewer side effects.
I'm definitely excited to see what kind of comes out of this research and everything, because speaking from experience in my family, a lot of us are veterans and everything, and so I would love to see how that could even further help them, some of the PTSD and everything.
Yeah, I think the current clinical trial, again, it's much more advanced for psilocybin, but the result so far has been quite exciting. Compared to current form of treatment, the most well-known and common one are Prozac and other forms of SSRI, which is the serotonin reuptake inhibitor. The psychedelic results tends to be a lot more rapid. So the response to the compound is a lot more quicker. Whereas for SSRI, you have to take them daily for several weeks before some of the effects would kick in. And then moreover, it's also much more sustained. So with just 1 or 2 doses, it seems like the decrease in the depressive symptom can be much longer. Again, whereas SSRI, that kind of medication, you have to be taking it every day. So there's a number of advantages that really excite the people in psychiatry. Although I have to say the current clinical trial are from smaller number of people. I think the biggest trial is around 200 people by Compass Pathway. And then there are some smaller ones at Johns Hopkins with about 50 or 60 people. So they still have to do that gold standard phase 3 clinical trial with several hundred or maybe even up to close to 1,000 people to really get that nailed down before I think we have further evidence. But it's very exciting.
I would be interested too, as somebody who has like medication-resistant depression. I've been on all sorts of different antidepressants, how people like myself, how the psychedelics would help more compared to like traditional medication.
That's where I see the main advantages is actually for treatment-resistant depression type of cases. Because actually the current type of treatment like SSRI or other forms like electroconvulsive therapy, they are somewhat effective. I mean, you cannot say they're not effective, and they're used as treatment, but they're only effective for a fraction of people, and there are a subset that don't really respond. And then I think the opportunity for psychedelic is that it has a very different mechanism of action. It works by targeting different brain receptors, and it's not really stimulating the brain the same way as ECT. So through the different mechanism of action, just the way it operates, I think there's a good chance that it might affect a different sector of people with different kinds of maybe even depressive symptom. At the end, it might just give the clinician another tool for treating depression that might another options in their arsenal, rather than straight up saying that, oh yeah, it may be more effective or less effective than SSRI. I think that's perhaps a better way to view it, and another reason why I think it's exciting.
I'm curious about these clinical trials, not to get too derailed. We'll get back to the paper in a second. But are the clinical trials for depression involving also like a therapist or someone guiding the person through the trip?
Yeah, so that part varies. There are trials where the role of the medical professional is minimal, as in they're there, but they're acting as more of a monitor and less so of an active psychotherapy role. And then there are other trials where the medical professional is much more active. So the case for the psilocybin is the one that's sort of less active, particularly the ones from Compass Pathway, which is the company leading the phase 3 clinical trial. And they quite explicitly try to have it so then it's more of a monitoring role. But still, you have to understand, it is still quite different from the current medication where you would maybe take a bottle of pill and go home and take it yourself. In the model of Compass Pathway, as I understand it, you will still meet the clinician before the dosing session to understand what you will experience and to talk about what will happen to get an expectation of it. And then on the day of, you'll be monitored by 1 to 2 medical professionals when you're being administered these compounds. And then finally, there also tends to be some session, maybe 1 or 2 sessions afterwards, to talk about what happened and to follow up. So there's a lot of human factor involved that's different than current type of medication use. And then there are other kinds of trial and other therapy that are, again, much more active, right? So the classic example for that would be the MDMA-assisted psychotherapy, which is actually even in a more advanced stage now for treating PTSD. So that's the effort led by MAPS. For these clinical trials, they have just completed the second half of their phase 3 clinical trial. So they completed all of their phase 3 clinical trial. I haven't seen the actual data yet, but the company said that the result has been positive. So there is hope that actually if it's really positive, as they said, then that could be sent to FDA for approval maybe within the next year. So that could be the earliest psychedelic related type of therapy that we'll see. But for MDMA, it's a much more active process where there's actually psychotherapy happening during the dosing session. Yeah, I think that the efficacy to which the assisted part, the psychotherapy part is needed, I think that's debatable. There's people that think it's very important. There are people that think they're less so. And I think overall, there's just not a whole lot of tests right now that go directly head-to-head comparison on whether they're needed or not. I mean, there's no doubt that psychotherapy alone can help people. It's just that whether if you add on the drug, whether that or if you do it the drug but without, like how do those different options compare? So for the first part of the paper, we did a behavioral study just to show that, like human, the drug is short-acting and then also can elicit psychedelic-like effect by this assay called the Hedgewood response. Then for the second part, we also want to look at other types of behavior because actually one issue right now with animal studies of psychedelic is that there's not a whole lot of behavioral assays that are robust, that respond reliably to psychedelics and related compounds. Many of the behavioral assays right now in preclinical studies, like with mice, are developed with other kinds of medication in mind. So, for example, testing SSRI, people would put mouse in a tank of water and see if they swim, or maybe they see if they prefer food with sucrose as sweet. But those are assays that are developed for, again, different mechanisms of action, like different sets of drugs. So it's not really clear psychedelic can work with them. So here we decided to test an assay called ultrasonic vocalization, which is another more innate assay. Here, innate, what that means is that we're not teaching the mice to do anything. We're just giving the mouse these compounds and then watch what they do. So these are more natural response to the compound. And in this case, the ultrasonic vocalization is a behavior that the mouse would elicit when they're in the presence of each other. So, for example, a male mice, when they're in the presence of a female mice, they would start to make these audible vocalization. And they're audible to mice, not to human. That's why they're called ultrasonic, because they're at a very high frequency range. And the male mice would do this as part of their courtship and mounting behavior, again, in the presence of a female. So in this study, you can see first we have two mice, a male mice and a female mice. These are grown-up adult mice in a small arena. And then we have a microphone attached on the top. These are microphones that can pick up ultrasonic vocalization. Actually, a good use of these microphones is if you go outside and you're very interested in the calls of bats. A lot of people use this to record bats around sunset. But here we're using it to record mice. And you can see that mice then make these very stereotypical vocalization, again, in the presence of a female. This is called male mouse. And so you can almost see there's like syllables. They would make them in short word, and then these vocalization have different shapes, and it's almost as if they're singing in some sense. Some people describe it as singing, although it's not as complex as that. The goal of this study was to see whether some of the psychedelics, including the one that we're studying, 5-methoxy-DMT, might also have an effect on this innate behavior, which is perhaps can reflect also social behavior, because this is a vocalization that happens during a social encounter. What we see is that if we train these mice to and habituate them to elicit this vocalization, and then we give them drug and then we test, what we see is that when we give the classical psychedelic psilocybin, you can see a reduction of about 30%, so they vocalize much less. When we give them a dose of 5-methoxy-DMT, this is now a dose 20 mg/kg, a dose that's informed by our earlier Hedgewood response study. You can see that the effect is even greater. It almost completely suppressed 100% these vocalization. So the mouse just stopped vocalizing, making these social calls. And then finally, we wanted to see if this effect was specific to psychedelics, whether it's very selective. So we also tested ketamine. And the reason was that ketamine acts on a different brain receptor, but then it's also used as a rapid acting antidepressant. So we give it a dose that we think corresponds to the antidepressant dose. And then maybe perhaps disappointingly, we find that ketamine also suppresses the social vocalization. So it doesn't seem like this effect is selective for psychedelic, which is maybe what we're going in hoping for, to find a behavior that can allow us to study psychedelics specifically. But still, I think it's interesting to see that these compounds, psilocybin, 5-methoxy-DMT, and ketamine, have such a striking effect on the ultrasonic vocalization.
I guess I'm curious, are these doses where the animals still have a certain level of like consciousness? Because I know at least ketamine is also used as sort of like a sedative dissociative, which means that like if you give mice enough of it, they will kind of stop moving around and look more like they are unconscious. So are these doses where the mice are still like look awake?
Ketamine is a fascinating drug. Ketamine was developed initially actually as an anesthetic, as you suggested. It was developed as a safer alternative to PCP back in the 60s. And it was used as an anesthetic and still does for certain cases, like for horses and in the veterinarian case, and also for sometimes for pediatrics. But that's at high dose, like you said. At slightly lower dose, but still a psychoactive dose, this is where it could act as a rapid active antidepressant. So here the drug is no longer anesthetic, but it has these rapid antidepressant effect. What's interesting about ketamine is there's also an acute effect with ketamine, right? So this is why ketamine is also used as a recreational drug. You can get dissociative effect. So it's more of people describe it as a out-of-body type of experience. There are also perceptual changes. So it's related, I think, to psychedelic effect in some dimensions. For example, the perceptual change, but it's different in other cases. I don't think psilocybin is as likely to elicit these kind of dissociation. So there's some overlap in the acute behavior. And then interestingly, ketamine also has these longer lasting antidepressant effect. So ketamine is much more developed. And then I think as many people know, 3 years ago, it was approved as a potential, as a treatment for major depression, or more specifically treatment-resistant depression. And that was the esketamine nasal spray. So now there's a lot of ketamine infusion clinic. So here we use it as a comparison again, because it targets a different brain receptor. It has some almost comparable behavioral effect, both in terms of the acute behavioral changes and then also the longer lasting antidepressant effect. So if you go on to the paper, we did a little bit more analysis. One of the interesting things about the social vocalization is you can also characterize different types of social vocalization based on just the sound of them. Some of them have a very simple sound, staying at kind of one pitch. Some of them are more complex, where the pitch would go up and then down. And some of them go just up, or some of them just go down. And here we actually apply a machine learning algorithm to automatically detect all of these vocalizations that the mouse have made and then classify them. And interestingly, not only do these drugs suppress the vocalization, they also shift the type of the vocalization that the mouse make. I think it's a little hard to see from the data because there's so many types. The machine learning algorithm identify 11 types. But overall, the general trend seems to be that the animal made less of the complex vocalization, and instead they made more of the simpler vocalization where there's only one pitch rather than some of these shifts in pitches. So it's a little unclear what this means. I think it's just one observation that we have noticed. I think the research on what these vocalizations signify for the mouse is not really clear. So overall, this part shows that in addition to the head twitch response that many of the research lab does, there is at least another innate behavior where these drugs, the psychedelics, can have a very strong effect on, and that's the ultrasonic vocalization, which relate to the social aspects of their behavior.
I'm just more curious of now, like, how that translates to people. Now that we've seen what happens with mice and their vocalizations, how that translates to, like, when people are using it.
Yeah, so for 5-MeO-DMT, again, people describe the experience as very intense, the acute psychedelic experience, much more so than psilocybin. In fact, it might be also a little bit more dissociative than psilocybin, and then maybe a little less perceptual for some people. So there are a lot of nuances in the subjective effect between the different psychedelic compounds, which I think is actually one of the more fascinating things about these compounds, because there are thousands of different analogs of psychedelics, and they each have just slightly different variation on the types of subjective experience they elicit. It has to do with the neurochemistry and how they act on the brain. And then here, the motivation for studying the social aspect is that these psychedelic also has been known to affect social behavior in in humans. A related compound, MDMA, is known to be quite pro-social, right? That's ecstasy. That's the one that we talked about on being on trial for treating PTSD. Psychedelic is less so, but also have some of those tendencies. And people will report to be more connected with others to some extent, but also to the world. So here, I think one of the thing that inspired us to look at this assay was it's harder to establish social behavior in mice. So they don't, talk with others. They don't hug each other. But instead, this is one way we can look at it and quantify it. And this is the type of social behavior that they do, where during courtship they would make these vocalizations. It's one of their more naturalistic behavior. Then I think going more forward on the next part, so the first two parts talk about some of the characterization of these compounds in behavior, and then really just trying to show that this compound is psychedelic and psychoactive for the 5-methoxy-DMT in mice that we can measure. And then I think the figure 3, the third figure for this paper, is where the bulk of the result is. And it's the most central finding of this paper, which is where now we really try to look at now the potential for this compound to elicit or enhance neuroplasticity. So to do that, we use a method called two-photon microscopy, which is a laser scanning imaging method. It's an optical method. So we have these optical microscope in the lab. And then what it allows us to do is to work with live mice and then image into their brains directly. So how we do that is that we would take the mouse and then we would replace the skull with a piece of glass window. And maybe that's because the skull tend to be very scattering with light. If we want to look into the brain with high resolution, then we want it to be have optical transmission. So we replace it with a piece of glass window. And this allows us to basically shoot laser into the mouse's head to visualize individual neurons at high resolution. Some of the images that you can see from the paper are some of the images that we take from the microscope. You can see the individual processes from the neuron. So in the brain, the neuron has a cell body, and then a unique aspect of a brain cell is that it makes a lot of connections. So it extends all these little processes out. Even though the cell body itself is only about 10 micron in size, the processes can go on for about 100 micron. So it sends all these small processes out, and then that's how the processes connect, and that's how the neuron makes a connection and talk with each other. So what you see there is that we can image these processes, also known as dendrites. And then you can also see on the dendrite, you can see the little protrusion, the little parts that come out of it, the little knobs that are called dendritic spine. And that's the exact location where the neurons connect with each other and make synapses. Each of the primary neurons can make on the order of 10,000 synapses. And here we're looking at just a subset of them, but we can see them individually using this laser scanning microscopy method. So what we do then is to track these individual dendritic spines or sites of neural connection over days. First, we image for a couple of times, 3 days and 1 day before we actually administer the drug. And then we would administer the psychedelic 5-methoxy-DMT into the brain. Or in another set of mice, we would administer a vehicle. So these are control mice. And then we use the same imaging method to go back to the same location and track it for multiple sessions afterwards, in subsequent days. And then we can see just by comparing the same pieces of dendrite and dendritic spines, whether the mouse have gained some neural connections, or maybe they have lost some, or maybe most of them actually stay stable. In fact, most of them don't change. You might not actually want your neuronal wiring to change so much. So what we found is that typically most of these connections are rather stable, but there are some that are being gained or lost. And the big question here is then whether psychedelic 5-methoxy-DMT can enhance neuroplasticity. Can we see it structurally? And then also, if it does, then what is the time scale of it, and how long can we observe this change?
Could you, because I'm still having a hard time, figuring out what exactly a synapses is?
Yeah, so in synapses is the place where two neurons connect with each other. Neurons are the brain cells, and then the cells need to physically connect in close proximity to be able to communicate with each other. And then that connection is the synapse. So normally in a brain, the neuron is electrically active, and then that voltage gets sent down to different parts of the cell. And then at the synapse is when it converts to chemical transmission. It releases a chemical called glutamate. And then that glutamate flows from one cell to the other, allowing basically information to transmit from one cell to the other. And then now the other cell knows the first neuron has fire and trying to communicate with it. And that's how information gets propagated within the brain. So if you will, I mean, a good way to think about it is that if you know where all the synapses are and what neurons connect with each other, it gives you like a wiring diagram of the brain. If you think of a circuit, like an electrical circuit, and how resistor, capacitor, and things like that get wired up to form a circuit board. Here you have the brain, and you have different neurons, which are the individual components, and how they wire up is through synapses. except that in the brain is so much more dense. Each neuron actually have 10,000 synapses, so the connectivity is very, very dense, and that's actually one of the remarkable things about the brain. In total, there's in our brain at least there's 80 billion cells, and then each cell in the cortex has about 10,000 synapses. So the amount of connection is great. And the power of this method, again, why we chose to study animal here, why not study human, is that is because we can acquire these type of high resolution images in the animal's brain. We can visualize the individual synapses in here as dendritic spines. Whereas in a human, the current technology, because we're human, we cannot do these type of a little bit more invasive experiment. You can use things like magnetic resonance imaging, like MRI. Another imaging method would be, for example, CAT scan. Those methods tend to have much lower resolution, which does not allow you to see these kinds of connections between neurons that we can see in the mouse. Yeah, so I think another value of this type of research is that you might have heard a lot of talk. For example, if you read Michael Pollan's book, How to Change Your Mind, other people, there's been a lot of suggestion that, I think psychedelic can change your mind. It can cause plastic changes. But so far, there hasn't been too much evidence that it actually happened, scientific evidence. So this kind of imaging study really allows you to see if that's happening, because you can go and see the individual neuronal connection and go count them and then say, is there more? Are there less? So it provide that level of evidence and proof that, and then the next thing is how much there is actually changing and whether we can manipulate that kind of plasticity.
Right. I guess that's an interesting question is, does this increase in connections happen everywhere in the brain or just in specific places?
We jumped the gun a little bit, I guess, but the next part was like the 5-methoxy-DMT does increase the spine density. But I think, yes, that is a very interesting question, because here we're imaging one particular area in the brain. Again, we're taking these high resolution images, so we're focusing on one area. We're not imaging the whole brain of the mouse. The area that we focus in is called the anterior cingulate cortex of the mouse, which also relates to the cingulate cortex in human. So that's the frontal lobe in the front of our brain, the related region. And we chose that region because this is a region that's known to be affected in depression. So if you do PET scan in human, it's known that you have a substantial loss of synapses or loss of neuronal connection in humans with severe depression. Because of that loss of circuitry in human, we decided to focus on the analogous location on the mouse to see if this drug has an effect. But it is a fair question to ask whether this drug might have effect on different brain area. The interesting thing here is that these drugs are always administered systemically, which means that they get to everywhere, right? Because for human, you would smoke it, it goes in your bloodstream and it goes everywhere. In a mouse, we inject it with a syringe and then it also goes everywhere. So it has the potential to affect many different brain areas. But then whether it actually affects the brain area would depends on the local neural circuitry, whether it has certain receptors or not. So here for this study, we did not look at other brain region to answer that question, whether it affect other brain region. But in a previous study where we studied psilocybin, we actually did look at some other brain area. In addition to the cingulate cortex, we look at deeper area of the medial prefrontal cortex. We also look at another area, the motor cortex. And we find that some of these plasticity-inducing effects of psilocybin was quite widespread. We can see signs of plasticity in some of these other brain regions as well. So again, I think the drugs act more widely than what is maybe being implied by this type of study, where we're just looking at one brain region. It's something to keep in mind that, I think it's a concerted action of these drugs on the brain circuitry that makes its effect, not on a particular region.
Earlier, you were saying that like it hits to the serotonin receptor in the brain, correct? Is that also in the frontal cortex?
Yes, psychedelic acts on different kinds of serotonin receptor. Turns out there are, I believe, 14 different subtypes of serotonin receptors in the brain. These different compounds, they bind to different receptors differently. So the most well-known serotonin receptor for psychedelic research is the serotonin 2A receptor. The reason is that that's the receptor that's now to be established as being responsible for the psychedelic trip. And that's for human studies. So the evidence is quite decent. So if you give a human another drug that would block the serotonin 2A receptors, before you give them psychedelic, they may no longer trip. You also look at the amount of these receptors in the brain and how available they are, and then correlate with how much people is tripping, how strong that subjective effect is, and the correlation also quite high. So there's good evidence that these serotonin 2A receptors are responsible for the trip. But there are also many other subtypes, and people think that the other subtypes could also be important. And it's an open question on which subtype actually contribute to the beneficial effect, whether it's the same subtype, the 2A receptor, or it could be a different subtype that might be responsible for some of the benefits. And yes, the 2A subtype, and then also some of the other major subtypes like 1A and 2C, they're all present in the frontal cortex of humans and also in mice. But they're also actually present in many other areas in the brain. I think that's why it makes it quite difficult to pin down just based on the receptor alone where the drug might act. Because serotonin itself, which is where these drugs bind to, serotonin itself is also an important neurochemical that normally exists in the human brain. What these drugs do is sort of just hijack the existing receptor system that's already in the brain to perform their function. So with the 5-methoxy-DMT, using this method where we can visualize the individual neuronal connection as dendritic spines, you can see the result here where what happened when we actually image the number of these dendritic spines over days before and after we give 5-methoxy-DMT. You see that fairly rapidly on day one, the first time when we image these mice after the drug administration, we can see a rise of about 15% in the number density. What this means is we observe more dendritic spines, which is an indicator of more neuronal connections in the frontal cortex of these mouse. This increase and difference from the vehicle group persists over time until really the last time point when we looked at, which is 34 days, about a month after the drug administration. So one of the questions that we had, if you remember early on, was that whether some of these shorter acting psychedelic might also have a fairly short plasticity effect, or whether they have any plasticity effect at all. It seems like the result suggests that yes, they do induce plasticity, and then furthermore, the plasticity can also be quite long. So it provides a indication that the acute effect and that duration might not be so much coupled to the duration of the long-term plasticity, the structural plasticity that we can observe. I mean, you have to remember, a month is actually quite long for a mouse lifetime. These mouse typically live on the order of about 2 years. I don't think you can translate it exactly like that, but for a mouse at least, it's quite a long time. So the number increase of about 15%, you can see it's still quite a long time after the initial drug administration. The second component that we looked at is instead of just counting whether there are more or less of these dendritic spines, you can also ask, well, how big were the dendritic spines? And the reason for looking at the size is because the size actually relates to the strength of that synaptic connection. And what that means is that how strong is that connection? So if the first neuron fire and have electrical activity, how much of that glutamate is released and then actually received by the second neuron? You can have a strong synaptic strength where if the first neuron fire, it leads to a big signal in the second cell, or you can have a weak synaptic strength, which is even though the first neuron fire, the second cell respond, but only weakly. So that there's also tuning in the synaptic weight and how strong that effect is. And then that size of the dendritic spine, how big it is, really relate to the connection strength. So one could also then suspect, and as we have shown previously for different psychedelic, psilocybin, not only can it increase the density, but it also increase the size of the dendritic spine and make them bigger and stronger. Here, again, to our surprise, it seems that these properties are not coupled. Although we see that the 5-methoxy-DMT, like psilocybin, increase the number of dendritic spine, we see that it actually did not change the size of the dendritic spine, meaning that it did not make them stronger. So they're more, but they're not necessarily stronger. So the plasticity that is induced seems to be different for these two psychedelics.
Would you say they're like the benefit towards having more of these spines than having like the stronger connections? Would you say it would be better for like having more of them or having like them be larger, or do you want like a good balance of both?
Yeah, so I think that get at a good question on talking about why do we want changes in dendritic spines and why do we want these things the connectivity to change in our brain to begin with. I think that then gives a sense on why these observations might be important and could be a mechanism for the beneficial effects. Normally in the brain, you probably don't actually want to see that much wiring change because you have your treasure memory and you have your skills and you have these things in your brain that are wired and you don't necessarily want to change a whole lot of it. But then there are cases where you do want to be adaptive. So when people look at adult mice and see when do these connections change, they tend to occur when the mouse receive a new experience. For example, maybe when they have new perception or if they're learning something new. For example, people teach mouse to do certain reaching tasks like motor tasks. And then when they're actually learning and gaining these new experience, and that's when you see a lot of these dendritic spine change and wiring changes. Yeah, so it seems to allow the neural circuits to adapt and be able to be flexible in the wiring in order to adapt to new changes. To think about why these changes in the plasticity might be important in a drug effect, we think this could be one reason why when people experience psychedelics, could be quite important for the subsequent benefit. So the subjective experience, a lot of people cited that as something that's very memorable and very important for their life. And there's also some clinical evidence to show that the intensity of that subjective experience also can correlate with some of the clinical outcome. So that part, that initial experience could be quite important for the benefit. So what this suggests then is that enhanced plasticity could be one way in which those new experiences are being integrated into the circuit and being functionally ingrained. So this heightened plasticity is really a mechanism to allow us to integrate that experience into it that then eventually, again, solidify that beneficial experience. So I think this is one hypothesis for why the enhanced plasticity might be important, and which goes hand in hand also, I think, with the argument then why maybe some psychotherapy during this time might be useful to couple with the enhanced integration. That's one, I think, hypothesis. But then there's an alternative hypothesis. I think with science, there's always different kinds of hypotheses. Other things is a bit more biological, which is, I mentioned previously that there's also evidence that there's synapse loss in the frontal cortex with people with depression. So there are also some people that believe that, maybe this has not so much to do with this subjective experience that people have, and maybe the trip is really not needed, but it's much more of a biological explanation that you started off with synapse loss just because of depression, and here you're really normalizing that loss by a growth of new synapses. So factors like stress and genetics and other things that predispose to depression that might cause us retraction of dendritic spines and a loss of synaptic connectivity. And here you have a drug that promotes growth and to replace some of those lost connections. And that's a much more biological explanation.
It's fascinating.
But again, I think that what's interesting is, we look at when these growth occur, which is the timing that's, I think, important, which I think speaks to when these drugs are really active. And then I think it provides a window, like the timing where suggests that maybe that's when any kind of psychotherapy might be the most effective, or any kind of conjunctive treatment might be most useful. And then the fourth figure for this is a relatively short paper. But in the fourth figure, the last figure here, instead of just looking at the number of the neural connection and also the strength of them, here we're looking at changes. Because again, here we're taking advantage of the fact that we can just go back and look at the same connection over time. We can also ask, okay, if we see there are more neural connection, is it because that there is a gain of neural connection, there's just more neural connection forming? Or is it because that the existing neural connections are a little bit more resistant to elimination? Because you know that normally there's also turnover within the brain, right? As we learn, as we adapt, there's a normal rate of turnover of these connections being formed and being eliminated. So you can achieve an increase, if you have more formation going on, or if you have less elimination, or maybe a bit of both. So here, looking across, we can see that really is the formation of new neural connection that's contributing to the new, the higher density, the higher numbers of dendritic spines that we see by comparing images that we can see from day 1, day 3, day 5, after the drug administration. We can really clearly see that it's not really the elimination of the dendritic spines or the connections, is really just the addition of new ones that we can see in these animals that contribute to the higher density of dendritic spines. And again, going back to that timing, which I think is quite important, you can see that increase in the formation rate is almost exclusively in day one. The density, the number increase is quite persistent, but that addition of the dendritic spine, a lot of it just happened right on the first day, and then they just stick there afterwards. The elimination rate does not really change, and then the formation rate really also go back to baseline between sort of day 3 and day 5. So the elevated type of plasticity potential, the propensity to form synapse is rather short, but then the effect it stays there for a while.
I was actually looking at the graphs while you were explaining that, and it's actually really definitely that first onset effect where it spikes on the formation rate was vastly different.
Yeah, we see the first day. This is also true somewhat for psilocybin. We also see an increase in formation rate and no change for elimination. We've also actually done a very similar study with ketamine, and this is sort of where my research lab have all started, where we initially, way back almost 10 years ago now, we started working with ketamine and trying to understand its action. And then about 3 or 4 years ago, we switched to studying psychedelic. We've also done a very similar study on ketamine. We also see ketamine have similar effect, although the duration is shorter than some of these psychedelic. So here I show you the density can maintain up to a month, but for ketamine is shorter. It's actually starting at about 2 weeks time point. We can see it start to fall off, but then the increase in number density is also driven by increase in formation of new spines, which is also within the first day or two. So yeah, I think that also is a segue to why I think people are excited about this research. It's also because of the time scale of the finding that we show. That increase in the neural connection, again, for psychedelic lasts for about a month. For ketamine, it lasts for about 2 weeks. Those time scales actually matches quite well with what people have reported in the clinic. So for ketamine, if you receive a single infusion, you have a efficacy window of about 1 to 2 weeks. Unfortunately, a lot of people then have to go back to clinic and receive a repeated infusion with the hope that over time, maybe you can start to space it out. And then for psychedelic, at least some of the preliminary clinical trials show that the benefits can potentially be a lot longer. So the fact that these neural circuit changes, the time scale actually matches with the clinical finding is intriguing. Because there's not a whole lot of things you can find in the brain that changes for such a long time. When you initially give the drug, a lot of things are changing in your brain. But in terms of long-term changes, these are some of the few things that you can really reliably observe that then again matches the time course of the benefits that we see.
That's awesome.
That is awesome.
You mentioned a couple of times that there's sort of two camps in how psychedelics are working to achieve this sort of long-lasting antidepressant effect. And one of them is that you need the trip, and it's this sort of like mind-opening and life-changing experience that then produces these antidepressant effects. And there's another camp that's very biological and saying like it's purely the increase in these connections between the neurons because of the interaction with the receptor. Do you fall into one of those?
Yeah, I think here I'm going to put on my scientist hat and just say I don't think there's enough evidence for me to say either way. I just have to wait to see more data. They both have compelling reasons. I think the camp that leans toward that subjective experience is important. A lot of those are clinicians, the people who actually give people psychedelics. This is a bit of a segue, but it turns out it's really hard to blind a psychedelic trial because you can give them placebo and the pill look the same, but the people just respond very differently. So the people know, right, a lot of times whether they have received the psychedelic just because of their acute experience. But the clinician also knows just by seeing the people's respond. And then a lot of these people that are actively participating, they really swear that initial experience is quite important. And who am I to argue? I mean, I'm not the one that actually have seen these transformative change in the clinic, right? Firsthand. And then the second part of the people that tends to think the psychedelic experience are not needed, or I would say more the neurobiologists and the chemists. And there's a number of compounds now, I think a handful of them, where you can start to engineer compounds that can activate the same receptor, the 5-HT2A, the serotonin 2A receptor, but without actually potentially eliciting the trip. Well, that's a misnomer because most of these compounds so far have been tested on animals. So I've explained to you the head twitch response. So when you test it on animals, on mice, they don't have the head twitch response. And then so many of the neurobiology group extrapolate to say that maybe they're not hallucinogenic then. As far as I know, they have not been yet tested in humans to really see if they're hallucinogenic or not. But what I'm saying is, again, there's new chemicals that purportedly are non-hallucinogenic, but yet they're serotonin 2A receptors. So I think it is a testable hypothesis now. And some of these compounds are going to eventually be brought to the clinic and then see whether they have any depressant properties. So maybe in 3 to 5 years, we might start to see some evidence. And there's obviously a lot of advantage to that. I mean, I think we should also talk about some of the limitations of this. I think that in the media, there's a lot of hype and a lot of talk about the benefits, but maybe less so about some of the caveats of psychedelics. And actually, as a result, why it might be useful to engineer out some of these effects if you want to use it as a treatment. One of the key issues with psychedelic is that it interact with the serotonin 2A receptor, and that's actually also how antipsychotic medication work, which is to inhibit these receptors as opposed to activate them. So you can imagine it's kind of pushing and pulling. So it turns out that psychedelic is also really just not suitable for anybody with predisposition to psychotic symptoms or any kind of manic episode. So any kind of family history or genetic predisposition for these kinds of mental illnesses, you really should not be taking psychedelic, which could exacerbate the symptoms. And then another thing is that peripherally, your heart also have a lot of serotonin receptors there. So it's also not really good to take a lot of psychedelics repeatedly. Many cases recreationally and also even in some of these clinical trials, you're not doing that. You just, and people don't really abuse it that much. I mean, you take it, but not frequently. But if you end up doing that more frequently, you could also have heart issues because there's a lot of serotonin receptors and it causes heart valve problems. So there are a number of these, concerns that I think tends to be glossed over by the media right now. I think the media always swing, right? Initially, these drugs are very dangerous. Now, like they're panacea and can treat everything under the sun. And then eventually we'll probably settle down to somewhere in the middle where people have to weigh the benefits against the caveats and the side effects. And then I think one way to move forward also to engineer different analogs. So some of these newer compounds, second generation or third generation psychedelics might be less prone to some of these side effects.
Like, you said like generations of sorry, I'm drawing a blank on the word right now, but the generations of them, like what exactly do you mean by that?
Yeah, when I said that, I'm thinking about these compounds that I'm studying right now, like psilocybin, 5-methoxy-DMT, or DMT that we mentioned, LSD is the first generation, in that these are compounds that were known since the 60s. Many of these compounds also tend to be naturally occurring. So then they have also cultural history of use dating back to a long time. And then when I think about second generation, or even, I guess, later generation, I'm more thinking of compound that people now have started to engineer. So what they would take psilocybin and they would modify the different side chains chemically to make related compounds, but they're slightly different chemically, so they have slightly different properties. So that's what I'm thinking. And you can see throughout drug development, this is a common thing, right? If you look at aspirin, it is also initially derived from a natural compound. That's a different compound, but they have side effects. So then you gradually engineer a compound to try to make it have fewer and fewer side effects, and then eventually, arrive at something that could be more widely used. And I don't see why psychedelic would be different. Although that psychedelic is interesting again because of the cultural aspect and the societal aspect. There are people that swear that I have to take the natural compound because that's from nature, and there are a lot of people who thinks that these compounds, again, because of the history, they have certain significance. But I'm more open-minded than that. I think they can probably be engineered and improved. Just because they originally exist in a mushroom like psilocybin doesn't mean that has to be the best version of it. I think it can be improved and engineered and changed. So yeah, that's what I meant when I say sort of the next generation of psychedelics.
It'd definitely be fascinating to see the future of psychedelics and how that evolves in that sense as well.
I know you're not a clinician. Do you have any insight into, like you said earlier, people are now or the media is now sort of sensationalizing psychedelics as a panacea. They can, they can treat depression, they can treat anxiety, they can treat eating disorders, they can treat literally name a psychiatric condition, and people have said they can treat it. Do you have any insight onto to what extent that is grounded in reality as opposed to like wild speculation, because everybody's super interested in these compounds right now?
I don't exactly know where all the claims come from. I mean, some of it I think is true, as in I don't think depression is the only indication. I think there's some good evidence for other indications that would be useful for psychedelic treatment. So we've already talked about PTSD and MDMA, and there's also some trial now looking at psilocybin for PTSD. Some of the other more advanced clinical trials that really show some striking effects are substance use disorder. So there's a lot of evidence for alcohol use disorder from work from New York University, from NYU. Also nicotine use disorder. This is like with smoking. These are the anecdotes is striking. Like there are people who would smoke for 20 years and then they would like stop. It's hard to argue against that kind of experience. So there are some, and then also I think another one that has some very exciting result is cluster headache. So these are repeated headaches that comes almost sometimes for some people during the same time in the day that are very debilitating, and there's just no treatment. So I think there are some indications that could be helped, and these are being actively tested. And then there are a lot of also other I think claims that are based on less evidence. I think the good thing is now there's a lot of different clinical trials to try to tease out what might be real, what might not be real. So I think we'll start to see hopefully some of these things get tested out and see. I think there's one driving factor would be I think commercial interest. There's a lot of startup companies and companies starting up to test for different indications, and each of them want to have a unique angle. So I think that could be one reason why it started to try to be applied to different indications. This is not unique to psychedelic, but I think it's been done a little bit more so just because the startup cost for psychedelic companies is slightly lower than other kinds of drug development. Because if you think about other drugs, you have to first establish safety through a lot of initial testing. But psychedelic is different. The psychedelic already exists. You already know what it is, and it's already decades of research suggesting it's safe. So you can skip a lot of that initial stuff or accelerate it compared to a normal drug discovery. I think that's why a lot of people are jumping in, because you have this early start. Although later on, it's just the same difficulty where you have to run these big trials. So I think we'll also see the industry part getting weaned out and shrink quickly. But I think, these are some factors that maybe contribute to the current state in the media, just why psychedelic is hyped so much.
Do you think that psychedelics are going to go in a similar direction as cannabis legalization, where it kind of went this medical route of first medical cannabis became legal in a number of different states, and now recreational cannabis is sort of following suit? Do you think psychedelics are kind of going to go the same way?
I'm not an expert in this, but I do think that the people that are pushing the medicalization and also the legalization are taking a lot of the blueprint from the cannabis side of things, just because there's certain similarity in that the cannabis is also controlled substance, and then there's legalization and medicalization issues, and then psychedelic is a very similar thing. So they're kind of taking that playbook to some extent and then bringing it to in different aspects. And then you can see the result of that is also similar, right? As in, like, we're taking the leaf of cannabis now, you can see that many cities and then even some of the states now have decriminalized psychedelics. And then it might take a similar route on the medical side as well, where even if some of these treatments are approved, for example, the MDMA assisted psychotherapy for PTSD, you might also see distinctions where that whole thing has to come as a package in terms of as an approved treatment. So then it doesn't really change the Controlled Substance Act and how it's classified. Yeah, I think there are kind of many similarities that one could draw. And you can see a lot of crossover too. We talk a little bit about the industry. There's a lot of cannabis companies that then get converted to psychedelic companies. After the cannabis, they make the money. And then after the cannabis, maybe now a little bit more steady state. Let's go to make more money over the psychedelic. Let's see some of that. Yeah, I think there are definitely similarities there.
I'd definitely love to see like how it like affects here on the East Coast, because the East Coast has been very, what's the word I'm trying to think of, like hard to get either kind of been starting up and everything, at least like medically practicing and everything from what I've heard, especially in Pennsylvania.
I think that's actually one thing that people talk about, like especially for MDMA, where okay, it might be approved soon, right? Given all the positive data, but do we really have that network of provider? Do we have that clinic? And you have all this assisted psychotherapy. Where do you train all these people, and how do you make sure they're adequately trained? It's just a very different model of care, and then also can you even get insurance to pay for it because it's so different? And if you can treat it in one single time, then what does it mean for other kinds of treatment? if you're a drug company, do you really want to have this? you run it a couple of times, you're done. So yeah, there's a lot of these questions that are quite disruptive and not answered.
Yeah, that was actually something I was going to ask about companies getting into psychedelics, is like how, because yeah, like you said, it's kind of you do a couple of treatments and you see a long lasting effect. It's not exactly the model that drug companies typically go for, where, their biggest, money making drugs are ones that you need to take sort of chronically. So do you know how that is working at all? Like how do they plan to?
Yeah, I don't know so much. All I can say is that right now, a lot of the companies working for psychedelic and developing drugs, the companies are either new or small. And a lot of the traditional pharmaceutical company, I'm sure they're aware, just that I don't think they have any overt sign of getting into it yet for I don't know what reason. Maybe they want to see more before they get into it, or maybe again it doesn't kind of jive with their existing model of how to make money. Yeah, and then also true that a company point of view is how to make money. So I think that's a very different consideration. Let's say that you can show that psilocybin with the kind of guided and the guided administration is demonstrated to be efficacious. Can you actually make that a profitable treatment? Like what's stopping somebody then from, particularly if it's legalized, go out and just go eat some mushroom themselves instead of buying this expensive drug from the company, right? So there's also issues about, how do you actually make money out of this, and how do you patent and so forth. For at least the classical, again, this kind of more first generation psychedelics.
I think just to get a little bit speculative, that might cause the paths of cannabis and psychedelics to diverge potentially, because I think that the, medicalization and more widespread use of cannabis sort of has a synergistic effect where, these companies that are catering to medical cannabis users and companies that are catering to recreational cannabis users, they kind of feed off of each other and oftentimes are the one and the same. Whereas if psychedelics were to become used as a treatment for particular kinds of mental health diagnoses, I think your point is really good, that like, why wouldn't you just go somewhere else where it's like less, because it's not only the cost that maybe these companies would charge an exorbitant price, but it's also very gate kept by it being within these clinics in the medical community, where not everybody has insurance, not everybody has the means to go to these clinics and get these drugs and even like find out this is an option for them. So I think that might, I guess I'm worried that might mean that that might make it less likely for psychedelics to be recreationally legalized, because potentially these companies that are catering to medical use will not want that competition.
Your intuition, I think, is correct. In fact, if you look at some of the people, the money behind some of the effort going against legalization are in fact by companies. No, I think it's actually, the intuition is completely correct. They don't necessarily, I don't think the interest is aligned. So there are a group of people that initially started these, a lot of philanthropists, where I think is aligned. Yes, some of the people wanted to demonstrate their medical use because they wanted to have it more widespread and be legalized. But over time, there are other people, particularly commercial interests, where those are not aligned.
That is a very good point, because that's one thing we've seen with like cannabis and everything. There's a lot of organizations like that are pushing for, like, say, I think it's SAM or something like that, where it's like in Massachusetts as an anti-cannabis organization, and there's like quite a bit of them funded by like certain corporations and everything.
I like to end every episode with a bit of a question for all of you. So for all of you, what is something that you are excited to see in this field going forward?
Well, I guess for me, it's just more of the medical usages that we learn and everything, and how that would work more and everything. I'm excited to see like the science behind it.
I'm definitely excited to see how we were talking about like the rebuilding of some of those synapses in the frontal cortex of the brain because of like long-term depression, and see how maybe speculative, seeing in the future if that could be something that can be used to help replace some of those and maybe reverse some of the negative effects of depression on the brain.
Yeah, for me, my lab will just keep following up on this research. I mean, here we're showing the structural plasticity. I'm hopeful that in the next 5 years, within the next 5 years or so, we can start to figuring out, what are some of the molecular signals that actually drive these changes, and it also gets a bit more specific in terms of like what are some of the cell types and what are some of the brain region, some of the details on where these plasticity are happening. So I'm excited to dive deeper into this process.
Great. Well, thank you all for being on the show today.
Thank you for having us.
Yeah, thanks for having us.
Yeah, thanks. It was fun.
Before we go, do any of you have anything to promote, any social media? I know Raven and Asmodeus, you are on another podcast. Do you want to tell listeners a little bit about that?
Yeah, so we like we kind of said when we introduced ourselves, we're the co-hosts of Smoking Out the Closet. We talk about the intersectionality between the LGBTQ and the cannabis communities, and life events going on within those two, important news updates. We talk a little bit about our life in Pittsburgh, stuff that's going on in the community.
Kind of some information like when it gets released, like some of the science stuff we find and everything, or like how things work as best as we can.
And like our perspective as people working in the cannabis industry and the medical industry in Pennsylvania. So yeah, we release new episodes every Tuesday. We are on all of the social medias in some form as Smoking Out the Closet, Facebook, Twitter, Instagram. We have a TikTok.
Very cool. Yeah, I'll link those in the show description, and you all should go check that out. And then, Alex, anything to plug? Where can people find you on social media, learn more about your research?
I have a Twitter account, which I post regularly. You can search my name. It's Kwan Alex C. I usually post some things about recent research, including on psychedelics. And I also post pretty fluorescence image of the brain sometimes.
That's cool.
That is one of my favorite parts of science, honestly, is pretty brain pictures. Well, with that, we're going to wrap it up for this episode. We've been discussing the paper 5-methoxy-DMT modifies innate behaviors and promotes structural neuroplasticity in mice by Sarah J. Jefferson et al. Our expert for this episode has been Dr. Alex Kwan, and our guests have been Raven and Asmodeus of Smoking Out the Closet. Our music is by Sam Brunwasser. You can find more of his work at soundcloud.com/visualsnowbeats. As always, you can download the paper and read the transcriptions at inplainenglishpod.org, and make sure to follow us on Twitter, Facebook, and Instagram at Plain English Sci. That's PlainEnglishSci. Thanks again for listening, and we'll see you next time for another episode of In Plain English.